Good morning everyone. I'm welcome you all to this 23rd day of CSIR online training. Before entering into the section, please let me know, am I audible? Can you see my slides? Is it visible? Please confirm, let me know through the chat box. Am I audible? Yeah, thank you for your response. Now let me start the section. Okay, sorry for the delay here. I met some internet connection problems, so only I'm delayed. Today the remaining part of 11th unit and the 8th unit we are going to cover. In this the remaining part of 11th unit is some topics are there, important topics. That is the geological timing. Geological timing means the scientists are having different type of period. They are met different type of animals, different the history of animals and the organisms developed. That is the sign. The the history of lives on Earth is called as the geological time scale. Okay, in this time scale we have eras and periods. How we can set the eras and periods? It's an this geological time scale. These eras are in the eras that is classified into the periods. Okay, this is the one representative image. These are all the million years before. Okay, 4,600 million years before our Earth is formed. Then 3,500 years million years before our oldest microfossils are developed. That is depends on the geological and the historical research report. Then 2000 million years before they found the first eukaryotic cells. Then 500 million years ago they found the leaf of Edicran. Then that is this apart the before period of the 2000 million years before that period is a water based is water based life. In that they are found some organisms in the water also. These are all found in the land, over the land. Some of them are found in the water. That water based animals or organisms are the archaeans. Then at the period of 580 million years before they found the uh, proteros proterozoic. Okay, then next 540 years before they found the trilobites. Then 530 years million years before they found the champions. And they water also they found the cambrians. Then 5000 years million before they found the first vertebrate. Before the 5000 years before they didn't find any vertebrates. At the period of the 5 million years before the earth they found the first vertebrate. This is the very important step um, in the geological timing of the life. Then 4000 million years before they found the extinction means there is group of animals are died at the 438 million years before that duration. In that 400 year million before they found the uh, or some animals that in the water life. Okay, that's placoderm, and at 367th million years before, they found the coiled, that is watery animal, coiled nautiloids. That before that period also, they found some mass extinction. So due to some environmental change or environmental disaster, there is a mass extinction. At 360 million years before only, they found the amphibians. This is the we are starting the biology from this amphibians, right? That many of them are no this many every this from this amphibians alone, they converted the structure, backbones, and form many organisms, many species in the world. These are the overall output of the animal, the geological time. This in this era, here they ask the what is the era. In that era, they divided into different periods. In that periods, they have the dominant animals. Okay, in this Cenozoic era, at the period of time, the quaternary and tertiary period, the mammals are the major animal. Okay, it's the latest one. And Mesozoic, that is in that Mesozoic era, they have 4 different animals, and that period is called as the Cretaceous, Jurassic, and Triassics. Okay, in that period, the majority dominant animal is dinosaurs. Then Paleozoic era, they found different stages, that is in that different stages different animals are there. This is very important because they ask in that period which animal is a very dominant one. There's Permian. In that period, that reptiles are very dominant animal. And Pennsylvania, that period, that amphibians are the major one. Devonia, that period, the fishes are the dominant. Silurian, sea scorpions are dominant. That Ordovician, that period, the nautilites are the dominant. Cambrians, trilobites are the dominant animal. These are all the periods in Paleozoic. In Precambrian, it's an first period. In Precambrian period, Cambrian period, they found the soft bodies. There is no period because there is unknown, that is that is too long years ago, so they are not divided. In that period, that is which is dominant means soft bodies. Is another explanation about the periods, eras, and key events. What is the key events means? In that era, Cenozoic, they found the human evolves, and Mesozoic, they found the in the Mesozoic period, the extinction of dinosaur is a major event. And Paleozoic, in that period, Permian mass extinction, and invertebrate become common in that which era? That is Paleozoic era. These 2 events are very important in Paleozoic era. Permian mass extinction and invertebrates become common. Then Proterozoic period, Archean and Haden. That is a Haden as a too long year back. That is totally called as the Precambrian. That period, that's the earliest life. Here you can see one word, EON. This EON is it's a total, it's an apart from eras are the major group. In between they have the period. In that period they having years. This is one. The oral eras are totally called as the EON. EON is in that we have the phanerozoic in that the word the phanerozoic, it's having three different era. In that three different era they having the nearly 11 periods. In that 11 either that Cenozoic is very important for the human evolve. Then Mesozoic, the extinction of dinosaur is happened. So what happens? It's another explanation. It's a well explanation. Here one thing you want to know here, yeah, fine. In the EON they having I said right, the different EONs, that is called as the heptane, arcane, proterozoic, and pH. In that period at the 542, that is the proterozoic at the end, and in that period for 540 years ago to 540 million years ago to at present, that is the called as the Phanerozoic. That Phanerozoic it shortly mentioned pH. Phanerozoic, in that Phanerozoic period we have three eras, that is a Paleozoic, Mesozoic and the Cenozoic. These three eras are divided into periods. In that period, in that period, that Phylogenetics, in that Phylogenetics they having EPOCH. This is another term. This is already very important. That is the group of it's a classification of period. The EON, the era is the classification of EON. It's a divided. It's in between steps, and period is in between steps an era, and the EPOCH is a divided part of the periods. In that we have the Phylogens, Eosins and Oligocens and Miocens. Here another one is Pleistocens and Pliocens. Here events which what happened in the particular period. I said in the Cenozoic itself human evolve happen, and the one more thing we want to know is the quaternary is a glacial cycle. In the tertiary we found it's some species, a divergence of the species. Some species Puna Exigobacterium, and here you can see the events, various events. Please remember these events. And here we want to know what is the groups of plant. We are having the phylogenetic tree, right? How they develop the trees, how we can classifying the plants. These plants are classified into different types: angiosperm, gymnosperm, pteridophytes and pteridophytes. In this it's another way, it's a vascular non-vascular division. In one vascular division they having two, that is the liverworts and mosses, and vascular they having the Flechnophyta, and in that they having the seed plants. It's seed plants Angioformatophyta and Coniferophyta. Okay, these are only common. This Coniferophyta and Angiosperm phyta, they having the flowering fruiting property. we have seeds and pollens, that conidophyta and angiospermphyta, they having the flowerings and fruits that are having seeds and pollens. And another one is tissue vascular lignin. This tissue vascular lignin found in the phyco phyta, conifera coniferophyta and angiospermophyta. Another thing that all the things are having the acidental green algae. Okay, these are all the classification of the plants. It's a non-vascular and vascular. In between, they have many different varieties, subtopics. It's a overall the image, another short forms here. The vascular, non-vascular, that mosses, it's a liverworts, hornworts, ferns, conifers, flowering plants. They comes under the first seed plants, and the first vascular plant. Here another thing is animal kingdom. Animal kingdom is converted into vertebrates and non-vertebrates. In vertebrates, they having the fish, amphibians, reptiles and birds, mammals. Mammals comes and all the it includes all the animals, seals, rodents, all things. In vertebrates, the that is different thing. There's they are in the they are having annelids, mollusks, arthropods, arachnoids, and echinoderms and protozoans. This classifications, whole classifications are the important. Why it's important? Once we want to classify the animal in the nature, means we want to know whether it is comes under which category. That is the thing we want to know. Okay, human evaluation, how we developed, what is the step-by-step process. It's a very important thing, because human evaluation is it's a very history key concept, key concept in the history. So we want to know the key concept. Here I will having 3 slides, 2 slides, right? First I will just explain the step process. The single cell, it comes to unicellular with the vacuoles, that is the converted into the chordata, and lobe fine means which are having the shape, then amphibia that having the that having the nature of the moving. It's only livable in water. It's having the nature with live in the apart from water, the land side. Reptiles, they totally can live in the land, and the forms are it's living only in the land, and the robustus, it's like monkey, and they formed the rectus and Homo sapiens. In that period, they developed. How they developed? The scientists are how would they said? Okay, this may happen. How they said that? They said which that their bones, the bones and the scalp, they collected the material and developed the content. Before the 4 million years before, in that period, 4 million years before, they found some the animals and the heads, due to which this head, they are having the different shape and different nature. From that, they collect the 3 million years before, they collect some material. That material is having some development in the previous year collected material, and that they developed the second 2 year ago and the 1 million ago year before this they found this scalps in between. That what that is the step by step process. Here Australopithecus, it's one in that they having two types. One is the Fagrilic hazels and Ethiopios. In that two things are forms the Homo naldis. In this Homo naldis, they are converted into the Homo erectus. This Homo erectus converted into the Homo uh, heidelbergenis. This Homo heidelbergenis converted that forms that develop to the Homo neanderthalensis, and its forms to Homo sapiens. These are all the eras which it's having the step by step process. There is they found the some not relevance between the in this second the in that period. These are all the things, but there is no correlation in the overlapping. But this fourth generation, 4 million years before they collected some material, that materials are overlapped. His overlapped means that are developed from the particular species. Here the overlapping fine. So that the final the three things are very important. Yeah, next thing is the neutral theory of the molecular evaluation. The molecular evaluation, how it is developed, there is a very hypothetical question how the molecular developed. It's from this question the molecular is the Motu Kimura, they explain he explained the concept the neutral theory of molecular evaluation in 1968. It's he observed two things. What is the things means the higher than the expected in if a natural selections were the evolutionary force primarily response for influence the level of genetic variation. Once the natural selection means it's a normal genetic evaluation that happen in the nature. This it's higher than the expectation. It's not in a control manner. It's higher than the expectation that makes the natural that makes the molecular evaluation. In that they having the two substitution. One is a synonymous substitution, another one is a non-synonymous substitution. Synonymous substitution, that is it's a similar same concept and two concept, and the non-synonymous is a not similar allele variants and the mixing of two molecules that is differ, that may happen or may not happen. Here the neutral theory of evaluation they explain with a sum. These kind of sums are maybe asked in the question, so only I'm included. The here the n is considered the size of population. They provided the size of population, that is n, with a neutral mutation rate at the locus of a lambda mutation per gametes per generation. Okay, that is, they provided the lambda. So they've asked the new what is the number of new mutations happen. They provided the mutation rate and population size. It's for this question the question is number of new mutations happen. It's a lambda into 2n. This is one output, and probability of fixation by genetic drifting. Drifting is it's a frequency. They ask p. How can it repeat? That question, if you want to answer p equal to 1 by 2n. 2n is you know the n is a population size. They provided the population size. Then another point of view, they ask the question the number of mutations, it's fixing with genetic drifting, the number of mutation into probability of the fixation. So by multiplying this, the lambda into 2n into 1 by 2n, so 2n become cancel. The output is the the lambda. So the number of new mutation per generation is likely become the fixed by the genetic drifting. It's a number of mutation in this value into probability of the fixation, this value. Here the two values are multiplied in the so lambda into 2n into 1 by 2n, so 2n become cancel. Your answer is become lambda. It's a neutral theory. Okay, next concept is a molecular clock. In this molecular clock, they have provided the hypothesis. It's a figurative term. It's technique. It's used to use the mutation rate of molecule to reduce time in a prehistory. Okay, here you can calculate the amount of genetic difference between the sequence. That different, using that different, you can find the you can predict the molecular clock, design the molecular clock. This clock is called as the evolutionary clock. The rate of molecular change is constant to predict the time of divergent. And so these are all the important things in the molecular clock. In that molecular clock, they are provided the different in the ascender. They having no changes in the gene, but 25 million years later they found some mutation at a particular point. At the same time they found the mutation at the various place. Okay, another 50 years billion later they found that mutation caused to two nearby molecule. This is the molecular clock development. Here the estimate the genetic distance, that is the D. Genetic distance means that is D, that is the number of amino acids replacement. Replacement means changed. Okay, this is the changed amino acid. This is a changed nucleotide. Then the using the paleontological data to determine the date of the common answer. That T is a time since divergent. How many times are taken? Then estimate the calibration rate. The calibration rate is the number of genetic changes expected per the unit time. That is R. The calibration rate R is the D by 2T. This value. So you can calculate the calibration rate D by 2T. Okay. How calculating the time of divergent in a normal sequence is the T, capital T equal to small d by 2R. This value 2R. So they provide the genetic distance. What is the number of amino acids are changed? This how many number of amino acids are changed, they provided in the sum, and the common aid center time they provided. You can find if you can find the question itself, or you can guess if there is a one amino acid change that happened in the 25 million years late before. After that, that take 25 million years for the one amino acid change. Then calculate for using that two time, using this two information, you can calculate at the calibration rate, or if they provided the calibration rate, or as the meaning of number of genetic changes expected per the unit time. With that value we can calculate the whole content for the divergent rate. It's a molecular term, the molecular clock equations, like it's nucleotide divergence between two sequences should be function entirely of mutation rate that k equal to 2 n mu by 1 to n equal to mu, right? You can cancel the 2n, so you got the mu value. That is the mutation. That is, here they mention the mu, that stuff they mention the lambda. The both are same. Here I want to tell one thing here, that time since divergence should therefore the reciprocal of the estimated mutation. The divergence, the time since divergence is the always reciprocal of the estimated mutation rate. That is the mutation, that is the lambda or mu, the t equal to 1 by mu or lambda. Okay, this is a molecular phylogenetic. In that molecular phylogenetic, we provided the morphological change, biochemical change, molecular change. Here this is a relationship finding thing. In that method they are used as the maximum proximity method and maximum likelihood method. Okay, what is the method based sequencing? Two different method is the maximum proximity. Find a phylogenetic tree that explain the data with a few evolutionary changes as possible. In maximum likelihood method, they found the tree that maximize the probability of genetic data in the tree. They won't explain the evolutionary changes, that find the maximum probability of the genetic data. In that direct method, indirect based method is a distance based method. In distance based method, they are the neighbor joining method, needle winch method. In this neighbor joining method, they found the tree that branches based on the branch length. Branch length is the relation. Close relations means they are having the close, very nearer and short length. This is the distance based method. And in the phylogenetic tree we have the 4 steps. One is the selection of sequence of analysis for analysis, then multiple sequence alignment. They did them in using the sequences. We studied in developmental biology how we can do the genetic genome sequence analysis. Using that method, they developed the multiple sequence alignment and tree building and tree evaluation. Okay, this is a tools which helps to understand the sequences. Okay, BLAST is a basic local alignment search tool which helps to compare the genes, the nucleotides or proteins. RASMOL, it helps to find the viewer. It's a view. We can see the nucleotide, you can see the DNA sequences. Here include some databases. The Unigene database, this is very important. They ask the question based on the data which present in the databases. Unigene having the data, it's an NCBI database which having the transcriptome information. Another thing is the DNA data bank of Japan, TDBJ. They provided the DNA informations. European Bioinformatics Institute, the EMBL, they having the all nucleotides similarity, sequence similarity, proteins characters, all things are present in the European Bioinformatics Institute. Then Swiss Prot and TREMBL, they both are having the protein database and protein information and DNA information. Here you can see which is specific for the which database is specific for which database. The nucleotide database having the GenBank and EMBL is specific for nucleotide. Protein database is the Swiss Prot and PIR. Structural database is the PDB, NDB, TALI and MST. Microarray Array Express. Chemical database PubChem. Pathways database KEC pathway. Enzyme database XPASI. Disease database is OMIM. There's a MIM, OMIM. The literature based study, we want to understand the concept, that is the PubMed based one or Scopus. Okay, how the origin of gene, how it where it is from. This is the gene is genes or new genes and proteins are produced for their functions and process. This source for the new gene or DNA is the poly polyploidy. Polyploidy means that duplicate genome can diverge. That is a diverse. That is uh, make a next generation one. Then gene duplication, transposition, retransposition, and import the DNA from organelles to horizontal gene transfer or horizontal gene transfer. Import the DNA from organelles means one organelle to another organelle change, or horizontal gene transfer. Then next thing, here I can see these are all the source, how that's the reason for the gene new gene production. This this is the example for the new gene production. Here you can see the chimeric gene that is the provided the chimeric gene. This may arise by the exon shuffling and retrotransposition. In that retrotransposition, first step is first example is the Jingvi, that is the Jingvi gene in the Drosophila. The first discovered example of new gene production is Jingvi gene that is present in the Drosophila. Two copies of yellow amber or that is called the YMP. It's arose by gene duplication. Here you can see the gene is present. The gene ADH gene retransposed into the introns 3 of the duplicate OMP gene. Here this gene, it's duplication of the OMP process happen. It's happened in the nucleus. Okay, then next thing is first after the gene transfer, that exons are during this process the exons are degenerated. Okay, this is a step-by process. First intron position is replaced with the it's duplicated, and the duplicate it's YMP1. That YMP is formed the new gene, the whole gene having the new property. The gene duplication and diversion. What is the duplication? What is the diversion? In this duplication, they having the separation, that different change, that is totally changed as like a sample. It's a sample 1, it's converted into same. There is a no change. It's a similar to the duplicated one is a similar. But in a divergent, they having the different change in the gene. Here you can see the change in the gene. Here rectangle, here there is no rectangle. The divergent means diverted. They have that gene having the similar property with other additional property. Another example for what is the duplication and divergence, and in this gene duplication they found it is as such converted into the same thing. Okay, then there is a mutation enhanced into A2 protein that produce after that mutation, using that mutation that produce a new gene. But in this duplication, this happening that such similar sequence are produced. Okay, and population genetics. In the population genetics, we have different terms. One is the gene pool, genotypic frequency, allele frequency, that things are very important. What is gene pool? That is a complete set of genetic information in the all individuals within the population. What all individuals in the populations means, if there is an elephant group, the all individuals that are all the same gene, that elephants are having the same gene, that is the pool, that is a group of the pooling. Then genotype frequency means the proportion of individuals in a population in a specific genotype. Okay, there is white elephants and black elephants. If there is white elements means there is a group of the if in that is a proportion. It's a 5, it's a 10 means that 1:2 proportion is there. So there is a genotypic frequency, how it is repeated in the population. It's differ from the one population to another population. Yes, right, as though one place to another place is differ. The allele frequency is a proportion of any specific allele in a population. Allele means it's a next generation. How much, how that parental, the how this one generation for that, how the gene is generated from one generation to another generation. That proportion of the any specific alleles. It's estimated from the genotypic frequency. These are only some problematic view. The 100 individuals in the populations, out of which the 40 is the homozygous dominant and 40 is the heterozygous dominant. Homozygous means that is a capital AA. Heterozygous means the heterozygous is a change. There is a one is the dominant. So capital A, small a. 20 homologous recursive. Homologous means homozygous. Sorry, homozygous means that is a similar recursive. Here dominant means capital A. The recessive means that is a small. So homologous recessive, that is small. Here you can understand the concept, right? That is the 40 homologous dominant. So capital AA. Dominant, so only they mention a capital AA. Heterologous, there is a change, that are having both A and B, that is small a and small a, capital and capital A. 40 is there. 20 homologous and recessives, that is small a. The frequency of A means dominant means that is the AA and small aa divided by the 2n, that is the total population. This is the value. You can please remember, capital AA plus capital A small a divided by 2n. That 2n is the total population into that is a n into 2 into n. Then the same, that is a small a, that is the recessive. Recessive means the small aa plus capital A and small a divided by 2n. This is the allele frequency, how it is allele is changed. Here you can see the GG, that is the dominant. Here it's heterologous, that is a small, both genes are there. It's a recessive one. This how it is allele with the next generation. How often it's shared with the next generation, that is the thing in the allele frequency, that is both alleles are going to change. Then gene frequency, in that allele frequency equation, number of copies of particular allele in the population and total number of alleles for the gene population. That is the number of copies of particular allele. One particular allele. This is the total number of all alleles. Then population genetics. In the population genetics is another terms I am included. That terms are called the different copies of genes, that is called as the alleles. That allele we have the A, small a. Capital A means that is the dominant, small a means is recessive. In that allele they are having three types. One is a homologous, the homologous having the capital AA or it's a small aa, and thing is heterologous as a capital A and small a. I'm explaining the individual concepts of gene pool in this topic. Here and then the examples, because the gene pool concept is they ask based on the genetic population. Okay, total number of genes present in in the individual of genetic population is constitute or the gene pool. So kind of genes present in the population, how it is contributed among the individuals. Okay, in the gene pool, first it's an example. In the wheat gene pool, the first generation having the gene that complete homologous genome. Then second generation partial or at least one genome in common, and third generation no homologous. How they said here, you can see that in the first generation they having the AA, BB, DD. This is the example of the nucleotides. Here you can see there is only AA, some partial. Then but here you can, in this generation there is no the gene. They mention ABCD is the gene. Okay, is there is a AA is a gene, BB is a gene, DD is a, there is a genes. There is no there is a partial appearance. AA is a partially appeared in this generation, but there is no gene. That is the complete. First one is first gen is a complete homologous. Second one is the partial homologous. At the partial genes are present, and third generation is a non-homologous. It's for example of which. Okay, here this is the gene population, genetic population explained by the Cody-Weinberg law. In this Cody-Weinberg law, that the diagram itself they mention. Here you can see that is the capital A, small a, capital A, small a. They find the draw. That is capital A, it's here. Capital A, small a, capital A, small a, small a, small a. It's the both are same, that we want to put the p square or pq. Then here you can pq get q square. That is a mentioned p. So equation of the all the equation is the p square is here, p square plus 2pq plus here the 2 pq's are here and q square. It's a equation of the Hardy-Weinberg equations. It's you all know that Punnett square equation. Okay, this is another explanation, and now we are these are the over things, major things in the 11th unit. Now we will enter into the session 8th unit. We take just 10 minutes for the 8th unit because this is too short and it's too informative. Okay, here you want to know Mendelian principle. In the Mendelian principle, they are having three different parts in that. Part they having the some alleles of dominant and cover of the recessive cells. The dominance, the law of dominance means the some alleles are dominant and cover up with the recessive alleles. In the segregation, how the law of segregation explains, an organism has two alleles for each gene, but they only pass one. They have a two gene, but they won't pass both. They only pass the one. That is a segregation. That is the segregation. They secrete one gene alone. Then law of independence are that assortment. In that the gene found on the separate chromosome are inherited independently each other. Okay, independent each other. Independently inherited each other. So this is the thing, the independent assortment, the gene found on separate chromosomes are inherited independently each other. This is a linkage map. Linkage map, how the genes are interlinked. This thing is a very important. In this, there in the map they quantified the genetic linkage. How they quantified the genetic linkage? The most hybrid test crosses and F2 generation. They based on the F2 generation output. Here you can see this is a P1. Here this, I think this image is not visible. Okay, I'll send a PPT, that time you can understand the concept. mm, the one gene having gray and the crescent and gray and crescent, this two is homologous. Here is a yellow and red, both are cross-linked, so gray and yellow, gray that crescent and red, both are cross-linked. This is the first generation. In that first generation, linkage is happened, and then you can for after the next generation, there is second generation, there is a change. In that change, they found the parental type. Okay, in that that recombinant frequency, the RF percentage, is calculated by the recombinant type into 100 by total number of the recombination happened. Here you can see the example of this image. Here first that the first linkage map is I explained with this. They are provided the sum, that is here no sum. They provided the example here. How they type, there is a no generation, no changes in the first generation. In the recombinant, there is a second generation happen in the second generation. For the frequency, they found this value, this using this equation, that recombinant type and into 100 divided by total. That classical concept of gene, in that gene concept is introduced by the Sutton, and they elaborated by the Morgan. That during that period, the Bitch and Miller also they explained the gene of concept of the gene. Because the earlier of the 90s period, 19th century, they are understand the concept of gene. In that gene concept, the genes are discrete particular particles inherited in Mendelian fashion that occupies the definite locus in the chromosome and responsible for expression of specific phenotype characters. In that, we want to understand some specific words, that's recon, muton, cistron, and complons. Here, what is a recon? It's the smallest unit of DNA capable of undergoing crossover recombination. So that recon is helps for the recombination. Mutant, its a portion is undergo the mutation. It's present in the DNA. That cistron, cistron is a unit of function, that is it's a gene in is a real sense capable of synthesizing polypeptides of an enzyme. The cistrons are helpful for polypeptide chains of an enzymes. Then amplicon, it's a unit of complementations. It's a change, it's a additional complementations. Then gene imprinting is a concept. In this gene imprinting, you can understand the concept of the preferential expression, of one parental allele. This one, okay, we can change, the allele can change. The epigenetic modulation mechanism that is happen in the gene imprinting, that is a GPG methylation. Here you can see the parental one changes the allele, and it's produce the it's a maternal one, it changes, it's a imprinting, gene imprinting happen. In this can with the example, here the it's a fetus, it's a ovarial gene. They provided the sperm from, and the male and female egg, separate the egg from the ovary, and first generation imprinting happen. So mat and pat is the maternal, and the parental one, they make a change in the embryo level, and they form the animal. Here you can see the both second generation, that is a maternal imprint, occur in the oocytes, and the second generation parental imprint is occur in the sperm. We taken the both from these two, that imprinting happen. This imprinting signals are example for the DNA methylation signal or histone modification signal. It's established during the gametogenesis process. That gene imprinting happen at the gametogenesis process. It's maintained throughout the lifetime of the somatic cell, and the germ cells of first generation, another round of imprint is resulting occur in the second generation. One is first generation, this may occur in the second generation. Okay. Then gene transfer techniques. In this gene transfer techniques we have, it's a machinery of a mechanism which helps to transfer of DNA from donor to receptor. In this, once donor DNA is inside to the receptor, as a recipient, its crossover can occur. How it is occur? It's having different method in the laboratory scale. In the laboratory scale, they are having the DNA transfer by calcium phosphate method, transfer of DNA using the polyphenol glycol method, polyethylene glycol method, and using the use of the DEAE dextron for DNA transfer, liposome mediated transfer. These are all the things happen in the laboratory scale with the chemical method. And electrical based method is the electroporation and electrofusion. These are the happen in the bacterial transfer. How it bacterial transformation is happen in the different three methods. It's a transformation, transduction, conjugation. In transformation, the donor cell break the wall and release the DNA, and that genes are enter into the recipient cell. That is the release of DNA. But the transduction happen with the this phage release. Okay, it's attached over the cell and release the DNA. That's the transduction. The phage induced donor cell, it's here, it's release a phage, and this phage attached to the other cell. In the one cell having the phage, that phage having the gene of the bacteria, the phage is released from the bacteria and attached to the another bacteria and release the DNA. This is a phage based transduction. Then conjugation, it's a direct transfer. It's attached, both cells are attached and transfer the DNA. Then pedigree analysis. This pedigree analysis was the most important one in the in your question. In this pedigree analysis, I'll send on some problematic video, because that is a very informative one. I found that and send you. Here you can see the generation, first generation. They are provided the a box and round. Here the second generation, that is Jane in the round, that is female one. Here that both correlation of the both produce the that occurrence of the change. That second generation change is one. This is very slow. It's two. It's partially in two females and fully in one male. Okay, this but this changes. How this changes may follow through the all, so it may pass through the next generation, or it may not be to pass through the next generation. It depends on the situation of the gene transfer. This pedigree in the pedigree analysis, we want to understand the concept, how the female characters and male characters, how they changed next generation. That is a very very important thing. The understanding of the concept is the percentage, how many percentage it's a transfer. And one more thing, it may partially transfer or it may fully transfer. Okay, the one more thing we want to understand, in that problems they found the fully darked one, that is the fully darked one is they call as that full dominant, the full auto, the fully dominant, there's a capital AA. Then that small small, that is non-colored one is a recessive one. Okay, that is the understanding each and every diagram having one explanation. We want to understand the explanation. That is very very important thing. Without understanding the this images, we can't go. That this darken, that red color coated one is called as the affected, and the affected, the without the that red color is called as the unaffected. These are all the terms involved in the pedigree analysis chart. Okay, in that kind of question, what kind of question means that if there is a inheritance of gene, it's a disorder, it's a how it is X linked or X linked recessive or X linked dominant. These are all the questions are there. In that question we can explain, because this here for example, here this is a normal one, but here this is a normal female, the normal male, that this is a affected one. Okay, this is partially affected one, and this is a X linked, X linked, because this female only pass this signal, so it's X linked dominant. These are all the things they provided the question here. The question means what is the possibility. It's a based on the concept of the affected, that affected and the non-affected. They ask many questions related to this concept. Here if we want to understand four things: X linked recessive, X linked dominant, Y linked recessive, Y linked dominant, autosomal recessive and autosomal dominant. This is the very very important terms in the pedigree analysis. And sometimes there is a triangle shaped one is also there. That is not that is also important, but in that case you can tell the they'll tell based on the image. Okay, there is a triangle shaped link between two compound, that is a two things, means that is a twin. Two triangle shape between two, this means that is the twin. Okay, the main thing is X linked dominant, X linked recessive, Y linked dominant, Y linked recessive, and the autosomal recessive, autosomal dominant. Autosomal recessive means that is a white one. Autosomal dominant means that is a change in color, that is changed. Okay, here the phylogenetic inheritance on color, that the concept is based on this allele, that pedigree analysis, how the changes are happen, that is a phylogenetic the polygenic inheritance. In this polygenic inheritance, at least four loci and possibly more genes are involved in this process. The example they provided, that each has two possible alleles, and that is the A B B C and 4. This is the two alleles, possible alleles. There's three loci, each having added to one unit dark and one lower case allele added nothing. That adds nothing means there is no added one. This is the concept behind the color chain. In the first step, first inheritance, there is no change. The 1 by 64, that is a 6 by 64. The change in second the second change, the final one is 1 by 64 again. This is totally the dominant. This is there is no change, that changes happen. This is the final overall the dominance. This is the recessive and two dominant. And what is the causes? It's how it is happen, how this color change, the polygenic inheritance, how it happens, caused by the mutation. This mutation happens with the chemical method or physical method or non-ionic method. In that chemical method, they having three various reasons. That is the nitrogen based base pairs change, alkylating agent, or acridinal acridine dye based change, or in a physical ionization change, that is X-ray, alpha ray, beta ray. Non-ionization means that is a UV ray based affection mutation. These are all the some point mutations in the DNA level. In the DNA level, there is a mutation happen on the T. Okay, this T change mRNA level is a T. It's a not change the amino acid, but it's a silent. There is a silent mutation. This is a no mutation here. It's a silent mutation. If there is a nonsense mutation is that it's waste one. So there is change in the first step itself, that change the mutation in the mRNA level, that stop the production of amino acid. There is a no chain, no proper codon is there. So there is a no link, no protein production. And the missense means if there is a change in particular position, that change the DNA level, that change the portion in change in mRNA level, that mRNA level after mRNA level change the production of amino acid change, that is due for the replacement of instead of lysine, here the arginine is produced. Or if the non-converting, that non-converting means there is no change in the here the arginine and the lysines have similar structure, but there is a totally different one, that is a conservative, here non-conservative. Then classical mutation having different type, that lethal mutation, sublethal mutation, subvital mutation, vital mutation, subvital, supervital mutation. What is a lethal mutation? When mutations causes a death of all individual undergoing mutations, whereas a lethal. And sublethal means it causes the death of 90% of individual, and subvital means it's less than kill the 90%. And vital mutation, it's when mutation don't affect the survival, that is a vital mutation, that not affect the survival of individuals, it's all are living. Supervital means this kind of mutations enhance the survival of individual. Okay, sometimes that's induce the production population, that is the this vital and supervital is the major thing which help us in the for the production of the insulin production animals and the plant for plant agriculture purpose. These are all the things used in the in the research. Then this is the final concept in the homologous in the unit 8, that is homologous recombination and heterologous recombination. In the homologous recombination, you can find two strands having the complex, there resections happen, there is addition of the RX or S2 complex, the protein molecule it's present there, that makes a stand invasion. That invasion means that there's a new change. Here this strand is going to start the new strand in the below one. There is this upper one is make a stand to the lower one. This two cross changes, the DNA synthesis and ligation happen. This two ligations are make a two DNA. That is a recombination. Homologous, the same genes are changed, that provides a two. And non-homologous, it's an DSP molecules, and as a chemical, it's make a recombination in the cut the DNA, and it's two DNAs are there, and these two DNAs are molded with that is edge attached with the Ku70 and Ku80. These are the that binding molecules, and it's clean up the end. It helps to clean up all the end. there is ligations happen with the both strand. The both strands are directly ligated. There is only one, the one DNA having two characters of the genes. Another thing is having the two DNAs. Okay, these are the things in the session 2. The two mark important questions are discussed. We discussed in the session 2. And tomorrow onwards we will going to discuss the session 3. The session 3 we have 4 units. In that 4 unit we will going to discuss the some important topics on the unit 1 that 1 mark the 2 mark questions also, that is less that not repeatedly asked in the exam point of view. So only we stop that session. Okay, that they provided the small time, the small period of time. Okay. If you have any queries, please post it in chat box. Thank you for everyone. Sorry for delay. Today I was met some internet connections, only I'm delayed. Thank you for your patience. If you have any queries, please chat it and post put it in post chat box. Ma'am, Javelin Ronnie ma'am, yesterday you asked some question. Actually, after the end of session you asked, so I can't answer in the chat box. I answered in the PPT. Please refer the yesterday PPT, ma'am. Okay, question session. I will go into question section. Okay, please explain TES or selfish segments under origin of new gene. Okay, how it is produced? This genes are the selfish DNA is sequence are having two distinct property. Okay, that two that two new genes product for new gene production, that DNA is spread by the additional copies of genome. It's having the that form, it's can ability to produce the additional genomes, and some another part is the not producing the that copy. Okay, that is another or one particular portion that not able to produce. So one portion having able to produce new genes. Okay, that new genes are it may affect it may face any chemical induction or genetical induction that helps that make a change, that may change the genetic information. That is the extra it's capable to produce the extra chromosomes, that is that the extra DNAs that are helpful for production of new genes. Okay, what is the significance of the pseudo alleles? The pseudo alleles are very important for the different expression, ma'am, because different expressions means it's a functionally related, but that having the different structures. It's a different expression of two genes, that's two alleles. It's they occupy the same locus, which divided into the sublocus means that copies having the different character. But they having the same locus, but sublocus are the different. It's in the pseudo they having the that having sublocus alone are changed, that also exhibiting the cis transformation. Cis transformation is the same structure, it looks similar, that's producing the changes, this pseudo alleles. And okay, next question. Is there any biological method for mutation? Biological method is that transduction, phage based method. That is the biological phaged method. Transformation, translocation, transduction, and conjugation. These are all the biological methods. The bacteriophage method is also used for the plant based, for plant gene transfer also happen with this bacteria. Agrobacterium tumefaciens, it's carries the gene in their nucleus, and it's applied to the plant, and that transfer the DNA to the plant genome. Okay. Okay, thank you everyone. Now I am going to end this live. Tomorrow we will discuss the session 3 with new topics.